developing and advancing methods for the analysis of high-dimensional heterogeneous data sets to identify and understand the association and the impact of nutrition and molecular information on diseases. In the big picture, she uses Bayesian approaches and machine learning techniques to investigate areas such as cancer prevention and detection, and as well as depression within children and teenagers. So I'm super excited to have her give her talk today. And Roberta, take it away.
Thank you so much for the introduction, Lauren. I think that was a very detailed, the most detailed introduction that I ever had. And thanks for the invitation and for allowing me to participating to this CCMB series. So today I'm talking about my work, my little adventure I will call, regarding my work that I started developing during the PhD and put the basis for other different researchers and the project that I am following right now. So first of all, let me give you an idea and a motivation behind the project. So as you can see from this picture, here we have different studies coming from different population. And in each study, we have some variable measured, and the variable are exactly the same across these multiple studies. So the difference between the studies are the patients are, and is of course the sample size. So let me give you some example in where in the data world you can find these this type of data. So you can find this data in microarray gene expression data. Here, for example, we have the breast cancer data, and here what we have is exactly the same gene expression measured across 7 different data sets. We can find these framework also in the nutritional epidemiological data, where you have exactly different population, but where you have the same nutrients measured across these different studies. And another example comes from the GTEx, so where you have different tissues and you can measure exactly the same gene expression measured in this different tissue. In all of these examples, the main question is how can we handle multiple studies from different populations? And so statisticians and data scientists try to answer to this question. And one really trivial answer and solution could be to stack, as you can see in this figure in the left, to stack all the study in one, to create this big data set and to perform standard techniques such as factor analysis or principal component analysis. So in this way, the result is that you can have common feature shared across all the study, but what about instead the study specific component? So let me refresh. I think that everyone knows what is the standard factor model, but let me refresh what is a factor model. So the factor analysis try to identify and to estimate the covariance structure of each variable in each study. And this is doable for deriving some factor component. This factor component have exactly 2 different pieces. So lambda 1, that is the factor loadings, and that of course has less component that I would call here J1, that is less than the observed variable. And then we have the L1, that is the latent factors. So lambda here is pretty important because it measures the influence that each latent factors, L1, will have on the observed variables. And finally, as the normal model, we have the model plus the error component. And this is the error component that is normally distributed with 0, with mean 0 and with covariance structure psi 1. So in the factor analysis, the covariance can be decomposed into one piece derived from lambda 1 transpose, that is related to the latent component. And the second piece, that is the second component, that is psi1, it's the error component of the error term. So suppose right now that we don't have just one study, we have S different studies. We can perform in each of these studies these factor analysis, and we can arrive to the factor loading matrix. The factor loading matrix generally is represented by a heat map. In this heat map, you can see the factors, you can see the genes, and so each rectangular here will measure the influence that each factors will have on the gene. So in this way, for example, in study 1, by performing factor analysis, you can find genes that are, for example, highly expressed. So you can find cluster of gene for factor 1 that are highly expressed or underexpressed. But right now, suppose that, again, we are performing S different factor analysis. You will have S heat map. So what can we do about this? So we want to arrive to some framework like that, where you can estimate this green part that, as you see, does not change from study to study. So it's reproducible, it's common across all the study. And then you want to estimate the study-specific component. So the idea is useful in different contexts, but it takes form from the analysis of genomic data. And building knowledge from this kind of data is a cumulative process which requires analysis that integrate multiple studies. And as a result of these multiple studies, some components are found to be reproducible, to be common across multiple studies. I mean, the main idea is that the genuine biological signal is more likely to be reproducible across different studies than the spurious one. But on the other hand, you have also the variation. So the variation for each study that can derive from the population variation, from a technical issue, like, for example, platform in which gene expression are measured that can differ from study to study, and so on. So in this genomic study, we want to capture this reproducible component and to remove these study-specific variation that can lack sometimes cross-study reproducibility. And for this reason, I developed the multi-study factor model. The multi-study factor model is a generalization of factor analysis. And what we have here, it's exactly like each observed variables can be decomposed in 3 different components. One that I called reproducible component, where you have the phi, that is the common factor loadings, and as you can see, does not have any S, so does not vary from study to study. We have a study-specific component that has a JS component, lambda S. So we will have for each study, one study-specific factor loadings. And finally, we have our error term. And as a consequence, this multi-study factor model can decompose the covariance structure in 3 different components. One that is the covariance structure just related to the common, so to the reproducible component. The second one that is related to the study specific, and the third one, finally, that is related to the error term. And this could be really important, as we see later on in this talk, because we will be able to, for example, build some gene expression network that are common across all the studies. So I want to show you 2 different applications for this multi-study model. So one application is coming from gene expression data. This is the curated ovarian cancer. And here what we have is 4 different studies with, of course, a different sample size and with genes that are in common across all of these studies. So the genes that considered here are involved in the immune system pathway. And the model choose just one common factor shared among all the study. And we also perform cross-validation to assess the prediction error. And this is a comparison between the multi-study and the factor analysis to see how much strength the multi-study can borrow by considering this joint analysis. And so the mean square error actually is smaller by comparing the multi-study for the two different factor analysis considering. So in the first one, I just merged all the study and I performed the factor analysis. So here we have just the common. In the second settings, instead, I just apply the factor analysis separately to each study. And as you can see, the mean square error is smaller for the multi-study factor analysis in these two different settings. So that's the output of the heat map for the 4 different studies. As you can see in the black rectangular, there is the common factor. And again, our model, the multi-study factor analysis, can estimate one common factor for this framework. And then we have also the study-specific component that, as you can see, are really different for each study. So these two studies are exactly measured on the same platform. And we also perform the correlation between the different factor loadings. And so as you can see, like the darker line correspond to the higher correlation. For these two studies, where the genes are measured in the same platforms, we have these 3 study-specific factors that are really correlated, highly correlated. So we perform again the multi-study factor analysis for just these 2 studies. And we discovered that these 2 studies has 3 common factors shared, as you can see again in the black rectangular. And we perform the Genset enrichment analysis to see if any of these common factors are enriched in one known biological platform, in one known biological patterns. And actually, we discovered that the first is related to the innate immune response, and the other 2 are not related to any other biological pattern. So this reveals that these 2 can be related to the platforms in which gene expression are measured. So this first example reveals you that the multi-study, first of all, has this strength, has this powerful to assess the common biological signal shared among all the studies, and remove also these study-specific variation that sometimes can impact the common biological signal. Another application that, we can see the multi-study from another point of view is the multi-study in these epidemiological data. So here what we have is 7 different studies. We have 4 studies from United States and 3 from Europe. And we have exactly the same nutrients. And like nutrition want to assess the dietary pattern, so the common dietary pattern across all of these population, but also study specific dietary pattern for each population. And again, here, the multi-study assess and estimate the 3 common factors. So the first, that the nutrition decided to call animal product and cereal. The second, antioxidant, vitamins and fibers, and the third one that is fats. Then the multi-study factor analysis estimate just one study-specific factor for the United States study. So this could be related, since the calcium is really high and also the niacin, to the difference in terms of breakfast than the United States can have respect to the European study. We also assess these factors with an odds ratio, with the probability of having cancer, specifically these oral and laryngeal cancer. So we ran a logistic regression and we control for other covariates. And we discovered that this animal product and cereal is positively associated with the laryngeal cancer. This antioxidant vitamins and fiber is negatively associated with laryngeal cancer. These fats has a sort of a contradictory behavior in the sense that for the oral cancer is positively associated, but for laryngeal cancer is negatively associated. And finally, just to study specific, Los Angeles and Boston are inversely associated to the same cancer site. So I wanted to show also this analysis because like we can see this multi-study to be applicable in many different consortia and to build some strength by the joint analysis of multiple studies. So right now, like we consider just, as you see, few variables, but I wanted to go in a high dimensional studies and settings. And I think that we are more used to see these sort of settings where you have like thousands of genomic variables and you have a small sample size. So generally, like we have a settings where P, the number of variables, are much greater than the number of the sample size. And so the factor analysis cannot estimate the parameter in this way. And generally, people add some prior or penalty to add some sparsity in the factor loading. So as you can see here, that's our factor loading matrices. And so if you can see right now, we don't have all the entry full, but we will have some sparse element. And this, I mean, allow us also to construct some gene expression networks. We will use these multiplicative gamma process shrinkage prior that is from Bhattacharya and Danson. Let me explain a little bit this prior, how it works. So we are putting in each element of the common factor loadings this normal distribution. So in this way, what we are doing is we are increase the sparsity as the column index increase. So if you are adding one factor, at one point you arrive at the solution where that column will be entirely zero. And this is meaningful because much information you are adding in your model at one point and your model will be super expanded. So you have to do a threshold to decide how many factors to retain. And we are putting also the same prior to the study specific factor loadings. So also here, I want to show you two different applications that has a very different point of view. So the first is to perform this Bayesian multi-study factor model in the breast cancer data. So here what we have is we have microarray gene expression data. We have 7 different studies from the curated breast cancer data. And of course, each of these study has a different sample size. We have different adjuvant therapy and we have like 10,000 of gene. So first of all, we retain the factors that explain more than the 5% of the variance. And so like the Bayesian multi-study factor analysis will retain 8 common factors shared among all the study. And of these, like they explain in total the 83% of the total variance. So again, let me show you the heat map. Here, what we have is the heat map of the 8 common factors. And I just retain the factors that were greater than 0.6, just for a easy visualization. And here what I did, I performed the Genset enrichment analysis again to see if each factor is associated with a known biological pattern. And what I discovered is that the first factor, so this one, is associated with the HERE pathway. And the HERE pathway is well known to be connected to the growth of breast tumors. The second factor is related and is associated with the cell cycle pathway. And the cell cycle is really important for cancer in general. So cancer, as you know, is an unchecked cell growth. And of course, mutation in genes can sometimes accelerate the cell division. So the cell cycle is really an important pathway in breast cancer and in cancer in general. Then there is the 5 common factors that is associated to the collagen. And this is really important and is related to the tumor progression in breast cancer. And the same for the 6th factor, that is ribosomal protein. And finally, the last 7th factor is related to the immune response. So the immune response, as you can imagine, sometime therapy for cancer can be associated to the immune response. So this again will enforce that the multi-study and the Bayesian multi-study factor analysis discover the biological signal shared among all the studies. We did also another analysis for the gene network. So the gene network is undirected graph. And we use the part of covariance matrix that is shared among all the study. And here what we can see is that each node is represented by a gene, and each edge represents an high expression between these two nodes. And we include edge between two genes if the common covariance matrix is greater or equal than 0.5. So that's the graph, the gene expression network that we found. And actually, we found 5 different clusters. So in each cluster, you can see the node. And the bigger nodes correspond to the more associated gene in that cluster. And you can also see that there are some lines that are blue and some lines that are orange, and that depends on the correlation. If the correlation is negative, that corresponds to the orange line. If the correlation is positive, that corresponds to a blue line. But this, again, this network analysis, again, reinforce the result that we have found before. And in fact, these 5 different clusters actually are associated to the same pathway that we have found before. So the HERE pathway, the cell cycle, the immune response, the collagen, and the ribosomal protein. Lauren, generally, do you do the question at the end, or if people want to jump in for any questions, I would be happy to reply in the middle of the.
It's probably a better Kelsey question, I imagine. Kelsey, you have thoughts?
Okay.
Okay. Well, maybe if they come in, I'll moderate for you. Definitely.
Perfect. Okay. Yeah. If there are any questions, just let me know. Another analysis that actually we did following the early gene expression clustering efforts, we tried to see if some of our factor is actually associated to these breast cancer molecular subtypes. And as you know, the breast cancer is often categorized in 4 subtypes. These are basal-like, luminal A, luminal B, and HER2 enriched. And these different subtypes present differences in clinical outcome and prognosis. So we first classify, for doing this analysis, the subject in these 4 different subtypes. And then we actually see the distribution of our common factors for these 4 different subtypes. For the first 4 common factors, we saw that factor 1 and factor 4 presented a different distribution compared to the others. So the factor 1 is really high for the patients classified in the breast cancer luminal A compared to all the other. And this factor is also present high loadings for some gene like GETA3 that are presented really high, especially in these breast cancer molecular subtype. Factor 4 is said presented lower score, as you can see, respect to the other 3 for the patient classified in HER2. And this factor is enriched in metabolic pathway. And this pathway has been found to be associated to this HER2. And finally, there is among the last 4 common factors, there is factor 7. And the factor 7 present a higher score for patient classified in the basal subtype. And so like this factor 7 is enriched in metabolic pathway. And these 2 metabolic pathways reveal an opposite association with the basal and the luminal subtype. So there are other factors that did not present any differentiation in terms of these 4 molecular subtypes. And in my opinion, this reveals the power of our multi-study to detect a biological signal actually that is important, but it's not captured by these breast cancer molecular subtypes division. So in this project, so for the breast cancer microarray gene expression, I just presented the output for the common factors. And this is because like I think that in the first analysis, we want to detect the common shared signal among all the studies, and we want to remove these idiosyncratic variation that can lack again this cross-study reproducibility. But there are any other framework where instead it's more important to detect the study specific, and instead it's important to remove these common characteristics. And this is exactly like the GTEx project, where here we consider tissue as studies. So all cells in human body contain the same DNA, but different cells in different tissue express on one hand a set of common genes, and on the other hand express another set of genes that can vary depending on the type of tissue. And in fact, gene in the human genome can demonstrate and can show diversity in expression level, and actually showing this tissue heterogeneity. And this is quite important because can elucidate the molecular mechanism of tissue development and function, and also tissue network can improve a drug target. And there are these 2 papers that actually are really important, where they actually show that gene and tissue specific patterns are more likely to become drug target compared to gene expressed everywhere. And also tissue network, it's a very important source of biomarkers for disease prognostic. So there are 2 considerations to be done. So the first is that oversimplified models can miss important tissue-specific patterns. And also, the important is to validate the result. And so to associate this result to a very important biological mechanism. So we are proceeding in 2 different steps. So we will apply the Bayesian multi-study factor analysis to identify this tissue-specific pattern. But in this case, we want to identify this tissue-specific pattern, and we want to remove the common component that can sometimes have an impact and cannot fully identify this tissue heterogeneity. We want also to validate this latent structure. And to do that, we perform this Eqtl analysis. And this has 2 aims. So one, we want to confirm that our study-specific tissue component is meaningful in terms of biology. And also, we want to provide important biological discovery. So I think that many of you know what is an Eqtl analysis, but just to refresh. So the Eqtl analysis is a very powerful tool to assess the association between a SNP and a gene. And again, we want to assess if our result has a biological meaning, and we want also to validate our result. So the SNP is a change in the DNA sequence. So suppose that we have this sequence, a SNP is a change in this sequence. And the simple way to perform Eqtl analysis is to perform a linear regression to test the association between a SNP and a gene. The problem is that if we perform the linear regression in each tissue, then there could be this common part that can impact and so can sometimes obscure the heterogeneity of the tissue. And so again, we will perform this 2-step procedure for the Bayesian multi-study factor analysis. So what I will show you here is first, I will perform the Bayesian multi-study factor analysis, and I will show you this tissue-specific gene network. Right now, I'm not anymore interested in the common component, but I want to remove this component to actually check and to improve the signal coming from this tissue heterogeneity. And then we will perform a regression model, but right now we are not considering anymore all the data, but we are just considering the part of the tissue specific that I am estimating from the Bayesian framework. And this is, this is considering the part that I estimate called the lambda S, that is the study specific factor loadings, and LIS, that is the latent study specific component. And we applied the model across 13 different brain regions. And the brain region is a very powerful method and sometimes reveal the change in behavior, and that can result in change in gene expression. So again, here, what I'm doing is a gene co-expression network, considering just this tissue-specific component, and each node will represent a gene, and each edge corresponds to a degree of co-expression. And we include the edge just if 2 edges has a correlation, a study-specific correlation greater than 0.5. So the size of the node corresponds exactly on how many co-expression this gene has in this cluster. So here, what I represent is the common gene expression network, and I reported among the 13 different regions, I reported just two of them. The basal ganglia and the hippocampus. So the basal ganglia is related to emotion and movements. The hippocampus is related to memory, short and long memory, and actually is one of the first region to be impacted by Alzheimer's disease. So first of all, I perform the Genset enrichment analysis to see if any of these factors are enriched in some biological pathway. And so both are related to this axon guidance pathway that is related to pancreatic cancer and Parkinson's disease. But then these two different tissue network has their own biological pathway. So the first basal ganglia is enriched in this signaling by GPCR. And this pathway regulates many different activities in our body, like from regulate hormone in the pancreas to the regulate these sleep rhythms. And the hippocampus instead is enriched in muscle contraction and calcium pathway. And this is one pathway related to Parkinson and Alzheimer's disease. Then I perform also the QTL. And of course, I'm not presenting all the genes found in this Eqtl analysis. I just show the genes that are presented in this network that are associated with a SNP. And actually, there are 3 genes in this network that are associated with a SNP, that are significantly associated to a SNP. And the first gene is related to visual memory. The second gene is related to Alzheimer's disease. And finally, there is the third gene that is related to depression. So this is another way to consider and conceptualize the Bayesian multi-study factor analysis in the term that here, as you see, I didn't consider the common and so the reproducible component, but we were more focused and on the tissue specific heterogeneity and how much this tissue can differ across each other by removing the common characteristics. So in general, like the multi-study is the only unsupervised analysis that can offer a statistical solution for this reproducibility. And we tried also to validate our result. So if you remember in the nutritional epidemiological data, we tried to validate the results on the logistic regression, or in this last analysis, we validate our analysis with the Eqtl analysis. And we can identify both a common and specific component. And this actually allows us to apply this method in many different applications and consortia. This is because like there could be many different aims, like to capture the common characteristic, to capture just the study specific characteristic, or to actually capture both. I developed this package in GitHub that has a Bayesian and also the first analysis that I presented, so just the multi-study, the simple multi-study. And also, like as you can see, the multi-study factor analysis is powerful compared to a standard factor analysis, because of course, like borrow strength across different studies and borrow strength from these jointly like studies. And I think that this comes to the right time, where we have a lot of different data and a lot of different sources where we can access the data. And so the reproducibility is not anymore one simple thing to think about it, but needs to be addressed in all of our biological application. And there could be many different future direction that actually I'm developing right now that could go in the environmental health or also in the methylation data. So I want to thank you all for listening. And I want to thank all of these people that work and have worked with me.
I'll go ahead and do questions, Kelsey, or you want to?
Oh yeah, I could. I just couldn't find my unmute button. So thanks so much, Roberta, for sharing your research with us. Are there any questions?
Thanks.
I have a question if no one has one. Roberta, for this type of factor model, how much power do you have here, or how susceptible to type 1 errors do you have when there's correlation that's shared between the common part across studies and maybe a study specific type of thing? Like you can imagine, I don't know if you have certain studies that maybe have certain like SNP chip biases that maybe other studies don't necessarily, or something like that, where you might have shared correlation, or if you have different environments or something that we have like this G by E thing that's kind of hard to disentangle. How well does the factor model kind of handle that kind of like shared correlation across different effects?
Yeah, I think that this it could be a nice extension, and I could think also to, for example, right now in the let me see, in the factor model, like I consider just one error for each study, but I think it could be nice also to have one error for a study specific and one error for a common. I think that it could be more related to a sort of a multi-hierarchical model. And you can also do a sort of inversion better of the study specific and of the common. So right now, one thing that I don't have, I cannot make these sigma phi and sigma lambda s invertible. But that could be something that I see really well in this, for example, data application.
And how subject are you to your choice of K for like the number of factors? Are you pretty robust to that choice?
Yeah, that's a good question. So in factor analysis, there is still an ongoing research on how many factors to detect. What I did is I did a singular value decomposition on these two matrices, so sigma phi and sigma lambda s, and I just retain the factors that explain more than the 5%. I think that also this technique, and it was pretty robust in the sense that for the simulation, it was pretty robust simulations and to many different data scenario that I consider in my simulation settings. But I agree that there could be many other different techniques to make it faster and maybe to do something like not in two different processes, but just in one, also with a different estimation. And instead, for the multi-study, like not the Bayesian one, so what I did is I just detect the number of factors with the AIC and BIC. So what was the best model? By running like, I don't know, 5 different times with one common factor, with 2 common factors, 3, 4, 5, and I just like choose the model with the less AIC. And I think that also there, especially there could be many other techniques that can be applied and not just the AIC and BIC.
I have a question. It's Amelia here. So I guess maybe related to what you just said. So could you get a sense of, I guess, do you have a sense, or could you obtain like a goodness of fit for some of these models? Like how can we know whether, I guess, yeah, that the model is appropriate for the data?
Yeah, that's another question. So what I use is, again, is the AIC and BIC criteria. So that's really a related question. I also, what I did is a comparison with the factor analysis here with the mean square error. Like, I don't know if, let me see, where is it? So here it's exactly like dividing like the data in training and in prediction. And with the factor analysis, with the multi-study, I predict these units. And so the mean square error was smaller for the multi-study compared to the factor analysis. that's the only goodness of it that I did. I perform also in this multi-study, so not in the Bayesian one. I perform also a likelihood ratio test together with the BIC and the AIC, but I think that this is quite in online with the AIC and BIC.
Okay, thank you. Nice talk.
Thank you so much.
Any other questions?
I have one. Hi, Roberta. So I have a question about your slide and comment about the tissue specific effects, and you work a lot in you know, like the different cancers, but not all cancers are part of the same tissue type. So when you look at more complicated cancers outside of like breast cancer, which you gave it as an example, how do you distinguish or differentiate between these different tissue types before you perform your analysis, or how do you integrate that piece? And I guess, you know, brain and spinal cord tumors are sometimes considered together, and then you get into the question of like metastasis formation. I'm not assuming you're looking at that at all, but once you get into, yeah, anyways, I was just thinking like how do you deal with that piece of it?
Yeah, you know, that's a very great question, in the sense that in the tissue example that I show, I didn't, so it was not related to cancer, but you are right. Like right now there is this, for example, another extension that I was thinking about is to this analysis, to apply the multi-study in this analysis called pan-cancer analysis, where instead of having different study, having this different tumor size, cancer type, thanks.
Yeah.
Yes. And so, for example, there we can detect with a sort of, I think like the Bayesian multi-study factor analysis cannot fully be applied there, directly applied, but one can find a very nice prior, and I think that it could be easily generalizable to this sort of data analysis, where you can find, for example, common gene across different cancer type and gene specific to this cancer type. I didn't think exactly to the different tissue there. I don't know if you have any.
Well, generally, like you pointed out, like breast cancer is just breast cancer. So,
Yes.
but there are other cancers that are more complicated where I think the factor analysis could be, your particular work could be super interesting. And the cancers are a little bit more complicated, especially those that metastasize quickly after their initialization phase. So I'll let you know if I have any ideas other than that.
Absolutely.
Other than just
Absolutely. Let me know if you have some sort of data or whatever. I was looking, I remember maybe some months ago, about these pan-cancer analysis, and that could be, I think, a very nice extension, because right now they are looking just a common gene or cancer-specific gene. So that could be a nice extension.
Great talk.
Thanks.
I also had a question about the cancer data, the head and neck cancer. It's just a very small detail. I was wondering, I just wanted to confirm that that was dietary calcium rather than calcium as measured in the bloodstream.
Sorry, can you repeat the question, because I didn't hear very well?
Yeah, my computer is overheating.
So sorry.
Yeah, the head and neck cancer data where calcium was identified as a common factor in the different geographic locations. Is it dietary calcium or is it calcium as measured in the bloodstream?
It's a dietary.
Okay, thank you.
Yes, it is. But the nutrients in these settings were exactly the same. So of course, just the population were geographically located in different area.
Thank you.
I think we've got time for one more question, if there is one. Good discussion so far.
I could tap one more. What's the scalability like for this, Roberta? And what's the bottleneck for computation, especially with the Bayesian model? Because I know obviously I know the prior pretty well, but I know it doesn't scale amazingly to like super large data sets. Are there any considerations that like people should think about here?
Yeah, that's also another great question. So first of all, I scaled the data before to insert in the algorithm. So the data are scaled. Second, like the ECM algorithm for the multi-study model is pretty good. The problem, of course, of the ECM algorithm is that you have to guess the starting point. And so there are many different methods to starting from a starting point. In our case, what I did is I developed first a factor analysis separately to each study, and I start the study specific from there, from that point, and it works quite well. For the GIP sampling, here there is a very big issue in the sense that the time point, the computational time is quite extensive. So also here, I was thinking to different extension, like one, it could be, and I think that it could work quite well to generalize these estimation to variational inference. And I think that this will work quite well, and it could make super faster the algorithm. Another is that I'm developing right now is actually to consider another prior and to actually do the EM algorithm. So perform the EM algorithm instead of the GIP sampling. But you are right that this prior is really good because it has a lot of different like characteristics, but it has some problem, especially when you scale the data in a very, very big dimension. With the Bayesian, I use this model for the 10,000, and for the tissue specific, I use that for 20,000. And with 20,000, of course, I have to use an high cluster computation, but it's impossible, yeah. Of course, and I had to use many different clusters to do that. So I think that with the variational inference, that could make a very big difference, especially if you are scaling with a very, very big data set.
Now that was 20,000 genes or SNPs or
20,000. Sorry, 20,000 gene.
Okay.
And people generally, I didn't have a lot of people and a big sample size. It was more the genes that had the differences. But I mean, the estimation is for any different genes. So the people, yeah.
Awesome. Well, thank you so much again for sharing your really exciting work. And thank you everyone else for joining us, and have a good afternoon.
Thank you so much. Bye.